So like Carl said, I have so our lab runs also a service, which is genome engineering, and we cover anything that comes to us from the campus. So we deal with all sorts of model organisms. I will limit myself today mostly to mice, but I will give you a little bit of introduction what we also do on the research side of the lab, which is we're interested in genome organization and ways to perturb not just genome editing, but just to change the organization of the nucleus in order to understand how the genome is organized. And the reason, what drives all of this research is our institute in general is interested in how cells build organisms. So it's from a developmental biology point of view. There is a interesting. The most interesting part of biology is this transition from the sequence of the genome to the giant diversity in organism shapes that comes through the complexity of development. And understanding this is impossible without understanding the function of the key players, which are in almost all processes proteins. And unlike other things like RNA and DNA, where hybridization is possible and it's like a generic method, for proteins, it is difficult to study individual proteins because by their own definition, each of them has different shape, charge, and everything. So there's no generic approach. And you could use antibodies, but it's difficult to raise antibodies to every single protein, and they're also not very good for live studies, so in vivo. And so the alternative that is now almost 30 years old is GFP, obviously, and all the other fluorescent proteins that came after. And through genome editing and variations of genome editing, we are able to attack any protein we want. So that's what our group focused on for many years, was basically to develop resources. So this slide outlines kind of like what the vision was and what we've been able to realize now for several model organisms, to have a collection where for every gene in the database there'll be a construct. And in this case, this was pre-CRISPRs, and we would use large genomic DNA constructs like bugs and phosmids as transgenes. And they're very good alternative to homologous recombination at the endogenous locus at the times when that was not possible. And even today, it's a quick way to assess the function of a gene. And importantly, it was also possible to engineer them efficiently through recombination, which you've heard about this morning. So I'm not going to go into details of recombination, just to say that we've developed methods to apply recombination in a multi-well plate to one plate is 96 wells, but we can process many plates in parallel, so we can do thousands of proteins. And once you have that, you're interested, say, in a particular transcription factor that's important in the nervous system, you will pull it out, make the animal, and then you can go on to study this is the developmental lineage of C. elegans in this case, and you can see which parts of the lineage is expressed, or you could do interaction studies, like in this example, it's a chromatin IP to study transcription factor binding sites, of which we also did quite a bit. So, like I said, the first model we started with was C. elegans, and this is the first one we managed to get to genome scale using phosmids. This is the pipeline. I will not go to the details because it's not the main point of this course, and also it's published. But in brief, you start with a phosmid and you end up with a phosmid on which the gene of interest is stacked with a fluorescent protein and some affinity epitopes. And also at the backbone, we insert a selection marker, which in the worms we could use to identify transgenic worms. And we did this on about 16,000 proteins for which there were phosmids, and we ended up with 91% of them correctly tagged and engineered through this method. So this resource is now available. We've applied it to many proteins to create lines and look at the expression of proteins. And these are just some examples of proteins where the localization itself is very characteristic of their function, and by that we could judge that they're functioning properly. We, of course, did many other controls, rescue mutations, and so forth. And the resource is performing beautifully. In this example, from this paper where we published the resource, this was funded by the Modern Code project, which was focused on understanding transcription factor function in model organisms. And this one was in worms. What you see in the movies on the left is in the development of the worm. Most of you probably know that the C. elegans has invariant lineage. So I'll play it again. If we start from the beginning, and we follow every single cell, just by following them and knowing which was the mother and which were the two daughters, we know the identity of every single cell. And at some point in time, you'll see in the second color here the transcription factor showing up. And by following the lineage, you know in which cells this factor is now expressed. And this allows you to create a map like this. You see initially there's 1, 2, 3, 4, several cells, right? And then each of them divides. There's a one cell division, there's another cell division, and then there's 2, and then there's 4, and so on. So by that, we can follow them, and also we can be quantitative. So we get this type of representation. We could also plot it as lineage maps. And so this was headed by the Robert Waterston lab in Seattle. And this resource is also available at this site here, and also all the transcription factor binding data that we achieved through ChIP-seq is also available from the Modern Code site. So the combination of these two things, so the binding site and the expression pattern, kind of gives you a fingerprint of transcription factor function in this model. We later adapted this same approach to flies, pretty much the same pipeline with a few details specific for the fly that are not important. This paper we just published in the beginning of this year, and these are examples of all sorts of different tissues. And the proteins beautifully recapitulate known patterns, and we discover many new patterns. This was done in collaboration with the Frank Schnorrer lab. He works on muscle development. And muscle is a very beautiful tissue because it's very structured, very organized, and just by the localization, you can exactly say if the protein is doing what it's supposed to do, and you can see all sorts of examples here, which I will also, for brevity, skip through. I just want to give you an idea of what the possibilities are. We've done similar things in mammals, obviously not in humans, but in human cell culture and in mice, using back transgenes. These are also examples of one of the first high-throughput papers in HUA cells. And this is just one figure from a paper which focused on the mitotic organization, so on the proteins that have a function in mitosis. And they were studied from 2 different directions. Resolution is poor, but the interrupted ones are from localization, co-localization, and the solid ones are from IP. So IPs were done for all the proteins, and localization was done for all the proteins dynamically through the entire course of mitosis. And basically, this is a summary of the entire process. So all of these resources are available through our website, where you can just conveniently go and search either by gene or by pattern that you're interested for, and you will come up with a list of genes and constructs, and or patterns if that's what you're interested in. And you can just search by. You can create almost quite complicated queries, because we have control vocabulary for life stage, for anatomic location, and subcellular localization. And a combination of this, you could ask questions like, tell me what is expressed in post-embryonic stages, in neurons, and it's nuclear in this case, and you get a bunch of proteins. Now, unfortunately, this would only be beautiful if we had all the proteins. We have a small number for now. But it is a start, and by making the resources available, for quite a while we were distributing them. Now they're distributed through a company, but they're available at a very low cost to anybody from the community. And by doing this, we just outsourced the difficult task of making all these wines, which we would have never been able to do all these thousands of constructs ourselves. And at least in the C. elegans world, people share their wines And it's very easy now for us to access all of them. All right, so then just about where we were wrapping up these projects, there was a small revolution happening. And that's when we switched entirely to genome engineering. And the rest of the talk would be focused on what you guys really want to hear about. As I said, we work with different model organisms, but I will try to stay within mouse. There are many ways to use Cas9 and all the other nucleases in terms of how do you deliver the protein. So do you put a plasmid, mRNA, protein, and so forth. What we do almost exclusively is protein and synthetic or in vitro produced RNA guide. We started with this around the time when we actually got ready to be seriously doing this. These papers started being published in which people did that. We tried it, we compared it with everything else, and we've been incredibly happy, and I can recommend it for any system because there are many advantages if you can use it. I mean, it's not always possible, but when you can use it, well, the first thing is the magic ratio. So what is this? It's when you use plasmids, you have often no control over the expression level of both the protein and the guide. And then they have to meet, and then people will report from different systems that, let's say you inject multiple animals, and one of them magically has a lot of the progeny is edited and none of the others. So apparently something happened in vivo in that particular animal that the ratio was just right, or they went to the nucleus at the very right moment, or we don't know what, but things work. When you have already the active compound in the shape of the protein and the guide, you can titrate that. You can put different amounts at different stages. You can find what, the Goldilocks zone, right? The other thing is species independent. For me, trying to work with many different organisms, this has been really useful. So you don't have to have specific promoter that works in that system. You can just use it. It's independent on the transcription state. So this is important, for example, in early embryonic development. In some systems, transcription is actually quite a bit delayed because of the material provided maternally. Transcription starts at, let's say, in zebrafish starts at 200, 300 cells or something like this. I'm not an expert, but until then, you're delayed. So your protein starts working way late, and that's not a good idea if you want to go germline. So another thing that might or might not be useful is that it's transient, and this is good in terms of off targets. I'll come to this point again later. I just want to make one brief point that it is very easy to make these guides in vitro. Did anybody cover this or previously? You actually did it in the okay. So I don't need to cover it, but you guys seen it's trivial. It's easy. We make a PCR and we just transcribe from the PCR. It's also now possible to buy synthetic guides, and we do that. There are several companies that are competing now. The prices are going down, so this is really nice. They're also chemically modified to be more stable, and the companies make various claims. I don't know to what degree they're relevant, but they're convenient to use. Okay, so the important point here, very important point, is that it is faster because there's no delay in terms of transcription, time for transcription, time for translation, folding, and so on. So you put it, it works. And in this example, from this crucial initial paper with the protein and guide, you have the left panel is protein, and this basically this assay here. If you see this band that is HA western, and there's HA on the Cas9. So you see the amount of protein. This is just normalization control here, but this band here is represents the edit. So the exact assay here, not so important, but anyway, the wild type is above. This is an edit, and you'll see with the protein is of course immediately as soon as you put it in. And this cell is cell culture. In this case, it's cell culture. But anyway, already after very, very short time, after 1 hour, you see at least a little bit. So in this case, this is percentage of Intel. You see 1% after 1 hour. And you wait a little bit, like 6 hours, and there's already significant amount in the cell culture. Whereas with plasmid, there was a big delay. And then I always wondered, right? You have this big delay, and in this case, it's just an indel. But what if you want to do repair? Your repair template, is it hanging around for 6 hours doing nothing, or is it rearranging and integrating in all over the place, right? So this is always a good idea. Then in this paper, they already claimed that this would also help with off-targets. They have on-target here, and they have an example with an off-target, which is intentionally there. And they see with the plasmid quite a bit of off-targets. So it's a bad, these are all bad sites, that heavy off-targets. And if you look with the protein, you seem to see a reduction here, whereas the on-target is identical. It's fine. The on-target is okay, but the off-target is reduced. The logic here being that the plasmid lingers for many days, and continuously there's Cas9 guide produced. Whereas if you notice on the previous slide, I don't know if I stress this, but it is gone on the next day. So the protein has some turnover, and then it's gone. Whereas the plasmid lingers 72 hours. There's still plenty, right? And how important that is another issue, because in my experience, off-target is not as bad a problem as it initially was feared, but it's still good to have this. Another thing that I also noticed helps very much in practice is that once you have the protein and the guide, this is already like a restriction enzyme, and you also have a template, because whatever edit you do, you have to have genotyping strategy developed as well. So that usually means PCR, and that usually means you did it before you started, so you have that PCR in your fridge. You use the protein, the guide, and the PCR, and you can see in vitro if it cuts. Quite a lot of the cases it cuts. So some people consider this unnecessary, but if you're working with mice where you have to wait 6 weeks to see what happened, it is quite useful to know that your reagent is functional, and we do it every time because it doesn't really cost much time or expenses. And you will see that often everything works, but there are cases, and in this example, everything works, but there are problematic ones. So this is like 50%, and there's some that are even worse. Often you don't have a choice, so you get a bad one like this. If it cuts even a little bit, I will use it, because this in vivo usually is enough to give us an edit. But we have seen some that totally don't cut in vitro, and of course they also don't work in vivo. Okay, so moving on. So this was covered, right? And you also did it? Did you guys do pronuclear rejections?
In vitro cut.
All right. So this is what we do a lot of. So pronuclear rejections, I will mention a few things from it. I mean, this is our cocktail. I'm not certain this is optimal. We're still changing it, and it works with a wide variety of changes. So, and it's hard to have satisfactory number of repetitions in mice. But I could tell you already that 10 times less than this also gives edits. And we have gone also up, but that's problematic because it's too much protein. But going down, the efficiency was not reduced significantly and might be beneficial to off-targets.
Mihail, your Cas9 protein, how many nuclear leader sequences?
How many what?
Nuclear leader sequence?
Oh, okay. So actually, I cannot answer this one for the commercial one we buy, because they don't review that. I think it's double NOS. We do have one that's produced by the protein purification facility of the MPI, which we have the sequence of. I could give you this with 2 NOSs from David Drexel, and this works just as good as the commercial, so we completely switched to it. I don't know if anybody can get it, but the protocol helped to express it and be functional and soluble and everything that's available. Yeah, you guys have that. So, is it the same plasmid, or do you made your own?
This plasmid actually is coming from US.
Ah, okay. It was surprisingly sensitive to adding tags to it and things like that. But we do have now proteins that work. Also, we have fluorescently. Yeah, we also have fluorescently labeled. We have Cas9 with cherry, GFP, and other fluorophores. Don't remember all of them. That we have the dead version of this, so you could use it for imaging. But
no, I was just curious because somebody used 1, 2, or 3.
Well, it works with 2. I don't know if more beneficial.
Yeah,
All right, so this is one example of an edit we did. What you should watch out into your results. So this is the wild type sequence. In this case, this is actually one of the clones, but it's wild type. And this is an edited clone. And in this case, we're changing this entire region. And you'll see that exactly the region where we're changing, it's a mixed sequence. And already from the double peaks, you could guess the correct one. Of course, you require topo cloning to be certain, and that's been done here. So this is how it usually looks. Most of the cases we get the heterozygote. Very rarely we get BLL conversions. And quite often, so this is heterozygote on top of a wild type sequence. But what we very often get is heterozygotes on top of an edited sequence that's non-homogeneously edited. So this is an example of non-homologous edit. The PAM is somewhere here, so it was cut here, and from here on, it's a double sequence. And you see this all the time. And what you should watch out is, your correct edit on top of a non-homologous edit can look like garbage. And we happen to discard quite some useful animals in the first round of experiments because we just didn't realize it. And then we topo cloned it. It was too late. But this is now all figured out. I want to give you an example of the efficiency. As I'll mention later, it varies a lot. So this is a best case scenario, but you can get up to that level. So in this case, this is the same edit, just more zoomed out. There are 2 oligos. We tried both of them, top strand, bottom strand. Here's the edit, and this is the wild type, and this is the mutant sequence. And we have 2 PCRs. As you saw, the edit was big enough, so we can put primers at that position. This is something I recommend in general, if possible, if changing. What we really wanted was 1 amino acid change. All the other mutations are silent. But this makes very easy to then genotype because you can place primers and you have a PCR that will only identify the mutant. And so this is now, let me see by the numbers here. It's 3 or 4 liters, it doesn't matter. 1 experiment from 1 injection. And you can see over half of them are positive for the mutation. This was unbelievable. And of course, when things are unbelievable, they're usually not true. There are already clues here. You'll see that there's some bands that are bigger than they should be. So that indicates problems. And so we've topo cloned then all of these founders. So we have multiple founders, and then we have 8 topo clones for each. And we sequence all of that to figure out what the situation is. And so what you really want is, let me see. Target. So what you really want is green, but what you often get is the targeted edit, but you'll get other mutations. And the yellow one is particularly bad because there's insertions or point mutations, and they happen very often. They happen in the oligos because they're synthetic. And they're really long, and mutations are inevitable. And unfortunately, that's been a problem. But some of them happen probably through a different mechanism because they happen outside of the oligo region. So what we've seen in some cases is that you get homologous recombination on one of the arms, and then no homologous, or homologous plus indel on the other side. So this is happening. So this first round, there were a lot of false positives, but at least you focus on the candidates, and then you identify them. Nevertheless, with 1 injection, multiple founders, unbelievably efficient. They also transmit very well in the germline. So in this example, we have 2 PCR. We can identify the wild type. This is the small band here. And we can identify the mutant, which is the bigger band. And this is a heterozygous animal control. And as you see, after crossing to wild type, you get what you expect, Mendelian ratio, more or less. And I don't have the data for that, but basically in none of the experiments, and we are already in hundreds of injections, probably not 1,000 yet, but hundreds of injections for sure. And none of the mice we made, we haven't yet met mice that didn't transmit. So everything has been transmitting very nicely. Also, what we've noticed is that we don't get mosaics very often. People have reported mosaics with plasmids and also with RNA for Cas9, mRNA. But with the protein, everything seems to happen either in the first cell or worst case scenario by 2-cell stage, because in the few cases when we've seen more than 2 alleles, there have been 3 alleles or something. We have not seen a lot of mosaic patterns. We're doing experiments now to determine this, exactly where it happens, and we don't know details, but this is the guess. It's probably the first cell. And if we look at the turnover of Cas9, it makes sense. It's probably gone by second cell division. All right, so this is an example of a more complicated edit we were able to do, showing what the potential is. People have demonstrated using multiple guides to do multiple edits at different genes. In this case, it's a little bit more complicated because it's multiple guides on the same gene nearby in the same locus. So this is within, let me see, about 5 qubit distance. We had to put 3 edits. So we had guides for each position and a repair oligo for each position. And we managed to get combinations of 2 very easily, and only one mouse that was a combination of 3, but still out of only, don't remember the number, should have written this down, about 120 mice, somewhere on that range. And unfortunately, that mouse was lost due to miscommunication. So we had to recreate it. But it wasn't not that difficult. We just took one of the mice with the 2 edits, and we added the 3rd one. Some lessons we learned from that project, which you probably encountered a lot. Many of these mutations, so we have to have them on the same locus, right? The heterozygous is not useful. We need the protein to have all 3 mutations. And we were quite happy to see multiples, these dark green ones, that appeared as homozygous. But actually, that's not the case. What happens is, in edits like this, is you get what you want, and then you get a big deletion between 2 or 3 guides. And actually, this is a separate story, but big deletions are very easy to create using just 2 guides. And then it will look wild type because it's just missing the binding sites for your primers. And when you genotype, you don't get anything from the wrong allele.
So these are small changes you're making?
These are all point mutations, okay. Point mutations. It's for collaborators, so I'll not go into what the project is, but it's not really important now. So in terms of general rules, I think you've gone through a couple of rounds. I might be repetitive. I just not going to give the basics, just a few things that I find useful. So it is crucial to disable the recutting whenever possible. By disrupting the PAM, or if that's not possible, the guide sequence. When you're editing inside of a coding sequence, this is relatively trivial. You just use a different codon for the same amino acid, sign and mutations, things are fine. We have had cases where we couldn't do that because the edit was of a transcription factor binding site and only one change was allowed. And we were able to get this from first try. So it is not impossible. This is not a must, but if I can do it, I always do it. So then another thing that really affects efficiency a lot is the distance between the cut and the edit. You should place the guide as close as possible to the edit, and you should compromise on a worse guide, but closer. That's what I would tend to do than a very nice guide that's, let's say, 50 base pairs away, especially with oligo repair. If you use larger templates, that's much less of a problem. But oligos and large templates operate through different repair mechanisms, and it is crucial for oligos to be close. Also, it's not possible to have oligos longer than 200 currently, so that's another issue. All right, so if you have to do 2 edits on the same locus, for example, the PAM, you want to destroy the palm of the guide, and then you want to make a mutation further away down in the oligo. I would highly recommend that you get mutations silent mutations in between, because when we didn't do that, we got a series of mice with both either of the mutations. So there's no preference for one or the other. I think the PAM mutation was favored because there's basically selection against it, right? But we could get both, and we also got the combination of the two, which is what we wanted. But when you change in between, that's treated by the repair as an insertion, and then you can only get both. And I think that's recommended. All right, so oligo repair efficiency depends on the strand you use, and I'll cover this in the next slide. So this is crucial, which of the two strands, because it's single-strand DNA, you can use either one. Which one do you use? And then it also depends on the length of the homology. There have been multiple papers exploring this issue, but I would recommend you read this one if you haven't, because we found that this influenced our results quite a bit. This came out in Nature Biotech end of last year. And basically, they claim that because of the mechanism of cutting of Cas9 and of the fate of the lesion after cutting, there is an intrinsic bias in efficiency, and their hypothesis is that they support it with some results. I don't know if it's finally proven truth, but there is data. So what they say is in the Cas9 guide DNA complex, one of the strands is engaged with the guide, right? This is clear. So the other strand then will be displaced, and you can see that in the structures that are now available. So upon cutting, this strand will be now displaced and more available to participate in homologous recombination than the other one that's engaged with the RNA. And that makes sense. They also show it for dead Cas9 mutant when there is no cutting. The strand is still more available because it's also displaced and available on the outside of the protein. And yeah, well, so that basically is the general rule. If I start saying now use the non-template or the one, basically think of this. One of the strands is available. Which one is it? Is the one that's not bound to the guide, right? So you would use a guide that will be that will hybridize to that available strand, and that will work better. But it's not as simple. So it also depends on the length of the oligos. I missed one point that's also important here. In the same paper, they actually showed that Cas9 persists on the lesion for a very long time. And this is something we also notice in vitro. When you cut, if you don't purify from the protein with PK, proteinase K, and then purification, you get smears, gel shifts, massive gel shifts. The protein is there. So they also notice in vivo, and they do single molecule experiments and so on. And basically, many hours later, they can still detect the protein, even after the cutting, right, with the nuclease. So it stays there, binds the two sides, on one side through the palm, on the other side through the guide, and persists on the position. That basically means that Cas9 is inhibiting normal repair through the usual, end processing and invasion and so on. But oligo repair will proceed because it has a template that's sticking out. So what they do is they tried the top and the bottom strand, and they tried different lengths of left and right. And you see this varies quite a bit. But there's one optimal case, and let me think now. So this is on the palm distal, palm proximal. Okay, so the palm is here basically, and this is downstream of the palm, and this is upstream of the palm. So in this case, that would mean that the strand that binds here is also the shorter strand, right? I think I get it. So if you design your oligo in this way, you have the maximum efficiency. But one other point that's also probably worth mentioning is that if you use much shorter guide, just 20 and 30, you get just half. Right? Still plenty of efficiency, and that means you could work with way shorter oligos. Yes?
Will this affect in the case where you use two different guide RNAs, like one.
You're talking like Nikkei's situation.
Or pair.
Or a pair of nucleases.
Like instead of using just one single
Oh, I see.
guide,
Yes.
two into different.
Yeah, they did experiments. It's all in the paper. They did dead Cas9 facing each other, just basically creating a bubble of exposed DNA, and that worked efficiently in vivo. So in the cases where off-target is really something you cannot afford, let's say gene therapy, using dead Cas9 is probably the best, the safest, safer than any case.
Because you could avoid this problem.
Yeah, that's what they did. Yeah, they used that. This was way less efficient compared to nuclease. So I would only use it when it is absolutely necessary. Let's say for patients, for humans, one day when we get there. But for model organisms, you can rather get the higher efficiency and deal with the off-targets by the many other ways you can, and I'll come to that. So in terms of on-target efficiency, when you're designing your experiment, you could pay attention to one of the many different sites and algorithms that allow you to predict the on-target efficiency. Unfortunately, I don't find them very predictive. So they're algorithms, but there is a good correlation if you look at the papers and so on. And in what we've done, there is some correlation. Like the better predicted guide also cuts better, but also we've had terrible, theoretically, guides that have worked fantastic and the other way around. So in practice, when you do homologous recombination, as I said, you're limited by what you have nearby, and you kind of ignore all of that. But it's still good that there are many useful sites that make the design easier. So I think the consensus from all these papers, the only thing that they seem to share is that stable seed of the guide contributes to higher efficiency, although that's also not black and white. So there was a paper in C. elegans actually, where they showed that a guide that ends with a GG before the NGG, so that's like a double PAM, works much better, on like tenfold more efficient than a standard guide. Or if they just move the guide one left, right? Almost the same sequence, but without an extra GG. So this is something good to know, but of course this combination is way more rare than this simple PAM. So this only has been useful for us in cases where we have to generate deletions or knockouts or something, where we have quite a bit of freedom where to put the guide. In terms of off-target, so I think initially the field was really concerned about this because there were a lot of experiments done in cell culture with persistent plasmids for many days, strong overexpression, and this appeared to be a serious problem. In vivo in the mice using protein, and also in other systems where you can use the protein like worms, people have actually in some cases tried very hard to find off targets and they cannot find them. So it doesn't seem to be that much of a problem, but still it's good to pay attention to in the design, use one of these programs to be aware of the potential off target sites. If your guide happened to be in a repetitive sequence, I will just ignore it. If I can, if there's anything else I can use, I will not use this guide. But other than that, it appears that even a single substitution at the first 10 nucleotides is enough to prevent cutting to a very significant degree, and it looks to be not that much of a problem. Protein and a guide, as I already covered, might be one way to resolve such problems. Another thing that came out in this very surprising paper that came in the beginning of this year was adding 2 Gs at the beginning of the guide, which they had to do because in this paper they used T7 produced guides. At some point earlier, it was noticed that T7 promoter requires 2 Gs in the beginning, as you've probably covered. So in earlier paper, they showed that addition of 2 Gs doesn't affect the cutting, and this we probably all noticed by now. But in this paper, they actually showed that it improves the problem with off-targets, or reduces off-targets. And this likely works by the alternative mechanism here, which was suggested in a different paper, which was to shorten the guide. And both of these cases, they basically will result in a less stable heteroduplex, RNA-DNA heteroduplex, than in a perfect match, by adding mismatch at the end or by shortening the guide. And because Cas9 undergoes structural rearrangements, so massive reorganization of structure upon binding and then upon extension of the guide, and finally adopts a final conformation that cuts upon full extension of the guide. There is an intermediate in between. Basically, this was transition is affected by this or that. And of course, it will affect more off targets than the on targets, and that's how it helps. I think that's the idea. So adding 2Gs to the end does not hurt in any way, so I recommend it in general because it shows that it helps. So how do we deal with off targets? I think the classic in genetic system when that's possible, obviously not in cell culture, but if you have animals, you will outcross eventually, and you're very likely to get rid of them by the time you have your colony of mice to analyze. But the easy way to do is to know the predicted off targets and just PCR resequence those regions as at least bare minimum. If you're working in cell culture or it's a very critical phenotype that you absolutely must prove that it comes from that mutation, you could go for whole genome resequencing. This is complicated and difficult then to distinguish background mutations from off targets. So there are many different methods that deal with this, and there was a nice review coming at the beginning of the year comparing the methods. And in my opinion, this is the best one, and also, of course, in the opinion of the authors, but they provide very strong data to support this. This method is called digenome-seq. The paper is mentioned on the previous slide here, Nature Methods last year. So it is really, really original and really efficient. So what they do is they'll take two purified DNA from two different sources. One is your wild-type control, and the other one is your edited control. So in your edits, you have, let's say, that's the insertion you want, and there will also be deletions and other substitutions. So when you cut wild-type sequence, you will have clean cuts at the position that's targeted by the guide. When you don't cut, you have basically no stacking of this sort. So what's happening? I didn't say what's going on here. This here, Illumina reads. Right? So next generation sequencing. Is everybody more or less familiar with? No? Okay. So what you do is you take the genomic DNA, you share it to fragments of defined length, and then you sequence from both ends. And in this case, you basically overlap. So you have the full sequence of that fragment. And then you map this to the entire genome. If the fragments are random, they should, like here, have no bias. In practice, there is always bias due to the sequencing artifacts and so on, but it's more or less a smooth representation throughout the genome. But if you have a specific cut and then you purify fragments of specific size, you will get things that stack around this cut, right? Because anything that was shorter is now less than the cutoff and it's gone, right? And this is very easy to computationally identify. So they plot here, that's more or less the distribution, and then the frequency of these cases comes as a sharp peak that's very easy to detect computationally. And now what happens if you have an edit? This is what happens. So you get reads around these positions that are identified by the cut of the wild type, and then there's some edits there, and these are also easy to computationally identify. They provide all the tools. You don't have to actually develop everything from the beginning, and the assay is very easy to do. And it's currently the most sensitive. And with this assay, they identified what I talked about previous, the 2Gs at the beginning help. All right, so with this, I covered like the general part about the mouse. And in the last part, I will talk about something we've been working on with the, if you want, Hutner lab. Yes, there's a question.
Regarding the detection of this off-target effect, in the case of milking, for instance, would you consider as well to do like southern blot, or would it be worthwhile?
Oh, that's different. Well, southern I can identify.
Because
spurious integration at other sites. And if you have a big insert that you can put a probe in, that's a very good idea. Yeah, it's not so easy when it's a point mutation or an oligo template, although oligos also integrate randomly at other positions. And that's definitely still the prudent thing to do, because things like that, integration at other sites, are very hard to identify. Also by methods like this, because you then align your reads to the reference genome, but you don't know what to expect, right? And you don't have your target there. You could do it if you want, but it is hard to identify events like that from pure sequence, and Southern is still the best. And it is a problem having your template, and also if you're using plasmids for the Cas9 and the guide, it is a problem that they will integrate non-homologous. It still happens, even though the homologous recombination is very efficient. Many of your clones might be contaminated with non-homologous edits, and by Southern you will detect them. And not by PCR methods might not happen, right? If you have a mouse and you have an integration on another chromosome, it will be fine. You will outcross it. You're okay. In cell culture, this could be a serious problem. So yeah, it's a good idea. All right. So in this last part, I'm going to talk about doing CRISPRs in vivo in the mouse brain. This is a project we did with the lab of Wilhelm Huttner, who is a director and a group leader in our institute, and this work was done with several people from his lab, primarily with Nereu Kalebic. So in neurobiology, a technique that has been used for many years, and it's useful for many different applications, is the in utero electroporation in the developing brain of the mouse embryo. So the procedure is roughly explained here. So the uterus of the mouse is a tube with multiple embryos. You can inject in the ventricle of the developing brain. At the early stages, these ventricles are rather large, and it's doable to hit them by microinjection without disturbing the further development of the animal if you do it right. And then you position the embryo between two electrodes and electroporate. And whatever molecule you put there, whether that would be siRNA or a plasmid, will end up migrating towards the electrode. And then by the way you orient it, you could send your reagent in different parts of the brain. And so this is very popular in neuroscience. For example, in this recent paper from their group, they are studying basically the big problem of the group is how do we have bigger brains than other mammals. And they're looking into proteins that are different between apes and us, and also between Neanderthals and apes. And they have, in this case, by comparing transcriptomes of chimps and humans, homed in on a single protein that is significantly differentially expressed in us. And then there's a plasmid here that they are electroporating in this direction in an earlier stage into the mouse brain. So now later, when the brain develops, you'll see that on this side there's a proliferation and they form gyri that kind of like us. Whereas the control on the left side is completely smooth, as it's normally into the mouse cortex. And this slide also illustrates one of the big advantages of the technique, that you have an internal control. You only electroporate a part of the brain, and you have the rest of the brain as a control. Of course, this is also a disadvantage if global effect is what you are after. And in that case, this technique is not very useful. But for studying cell biological processes at the level of development, this is actually really, really useful. So this was the first experiment we did. In this case, we have a plasmid as a source of Cas9 and the guide RNA. Everything is on one plasmid. After the Cas9, there is a reporter. That's a red fluorescent protein. In this case, here is in magenta. And you can see where the Cas9 is. This is actually not, the Cas9 is not tagged. There is a T2A FP. So they're two separate proteins, but it indicates that cell expresses it. So this, if we look at the overlay, you can see here this is the ventricle. This is where it was injected, and by this here white interrupted line roughly tells you where the electroporation went, and you can figure this out by the direction of the paprika expression. So you know which cells got the plasmid. So in this case, the mouse is expressing in this particular part of the brain GFP under a promoter that's specific for that region. And in the control, there is a guide that's not to GFP. It's to WAGZ in this case. It's a control should not affect, and as you see, there's no effect. But in this case, there is a guide to GFP, and the signal here is reduced. It's not very easy to see in this projection, but it is significant, as you'll see from the quantification later. And this is the second experiment we did. I mean, we of course repeated all of them many times and so on, but the second round of experiments, which were with a protein and a guide. And in this case, because the protein was not labeled, we co-electroporated a plasmid expressing cherry. And here it's very easy to see where the protein went in and how it completely obliterates GFP at this region. Now, in the quantification, there's a caveat here that it's not always the same cell that receives the protein and our reporter plasmid. So in the future, or now, what we're doing is basically using labeled protein instead of co-expression. Okay, so coming to the results. And
Sorry, did you use an antibody against Cas9?
no, that's not an antibody. That's this is now fixed sections, and the fluorescent proteins. This is the fluorescence of the fluorescent proteins. They survive the fixation.
This problem of tagging the Cas9, you use an antibody to tag
No.
antibody.
Oh yeah, we could use an antibody to Cas9, yeah, and then we will not need the reporter. I see what you mean. Yes, we could do that. But we also have labeled Cas9 that, with 404 that works just as good.
With that
Yes.
How many days you wait till doing the
This is
calculation? 2 days later?
This is 2 days later. We also did a time course at different time points to find out where it's optimal in terms of detecting it, and efficiency. 2 days was, and all 2 days was also what they were interested in because they care about this stage, so they had to inject 2 days before the stage they care about. I mean, in this case, it's just a reporter, but as you see, this is the quantification. As you see, the protein looks a bit worse, but this is because of this issue with the plasmid not maybe going in the same cells as the protein. We think now that the protein actually, and that's how it looked from the primary picture, that the protein is better, but it's normalized here to the cells that express, so that's why it's biased. All right. So this is all very well, and it's GFP. Does it work for a protein of interest? And so this is done now through sectioning and staining for a protein called TBR2. So we have a guide to it that should destroy it, and we have a control WAGZ guide. And as you can see here, the protein is nicely expressed in cells that have Cas9 expression, but it isn't. Or rather is look here, so Cas9 is expressed, but there is no TBR2, so and the quantification is on the right. So we managed to knock out TBR2 in about 50% of the cells that express Cas9, which is quite nice. And we quantified this, and well, it's semi-quantitative. It's not the perfect assay, but what we did is we will PCR around the junction, and then we will do deep sequencing of those PCRs. Now the caveat is that if there is a deletion bigger than the PCR, we will not detect those, and also it's still high throughput technique. So what we did is this is some examples, but we basically deep sequence these PCRs, and we get a lot of noise from the deep sequencing and the PCRs and that sort of stuff. So it's hard to judge what's an edit and whatnot, but it allowed us to look through what the main classes are. And the most common, and so this is it's all good news. So deletions are common, and they would often be disruptive to the gene, right? But the other thing, and insertions, so big deletions were actually quite common. and short not so much. That's interesting. But the other main classes that we saw was insertions, a lot of insertions, and also mismatches quite a bit. But insertions, that's why here is really interesting, because we get a lot of single nucleotide insertions right at the cut site, and as you can see, almost exclusively we get single nucleotides. We get a few that are 2, and not so many that are 3. And of course, 1 and 2 are out of frame, and that's very good news, right? Because if we are getting 3 nucleotide deletions, then that would often not be mutagenic, and explains why it works so relatively well. And since that lab works on many different neurobiological techniques, they have another thing set up in the lab which they can do very well, which is to use to section a part of the brain and then place that part of the section on a grid, embedded in a gel, where they can keep them alive for, I think, almost 2 days, and they will develop similarly to in vivo, but that allows them to have continuous imaging throughout the process. And on that type of culture, you could also microinject. So what you do is you take your needle, and actually without seeing anything, you aim at the surface of the layer where the nuclei are. There's no staining, there's nothing not to perturb it, and you inject. And you do that around the tissue at multiple positions, and some of these things hit a cell without destroying it, and you have a co-injection inert diet, so that you can identify them by the staining, a neuron that received the injection. And it works quite well, and by this method we also delivered the same reagent. So in this case, you'll see this Cas9 and a guide. There's the control guide again, and the GFP is not disturbed. And then there is the GFP guide, and very nicely, wherever we put it in, it's gone. Right? So that's good. The dye here is, I think it's just, what is it, Alexa or something like this, a couple to dextran. The dextran Alexa 555, right? So that allows us to just see which cells were injected. That's the principle. So moving on, we then did it also for the protein of interest, and we could identify it. When you knock out that protein, you see a basically defect in the development, which we were also able to specifically detect in those cells. So this was all very nice. At the time when we did this, we were one of the first labs that was that published that sort of thing. But we were obviously not the only lab working on it, because there were many other papers, and many of them now, well, not many, some of them were much better than ours. So I would recommend you to read this one if you haven't seen it. It's called, the technique is called Slender. It basically uses the same exact things we did, although they didn't use protein. It would have been even better if they used protein. But they moved it to a step further, which is what we are also doing now, is they did homologous recombination as well. And it worked very well. So in terms of efficiency, it's not very efficient. It's in the low percentage. It's around 1%. But percent of thousands of cells, it's still hundreds of cells. And actually, in the brain, it's very useful to have sparse staining. So what they managed to do is they managed to insert small epitopes and up to and including GFP. And then they either do staining or they do localization. And because it's not very efficient, it's not every cell. And when you have one cell out of 100 or hundreds, you see the cell nicely isolated. There is not really an example, but if you imagine, it will look something like this, right? There would be a single cell on a black background that is labeled. This is beautiful, because if you have the transgenic mice and all the cells were expressing, you can't see anything in the mess. Then you need something like a brain ball, if you're familiar with this technique, to be able to segment the cells and so on. Then a lot of image processing and so on to identify the individual ones. All right. So this is really not new. In neurobiology, they often use this sparse electroporation of diluted markers or viruses and so on. But this is now done through genome engineering at the endogenous locus. And they also show that they could combine that with a knockout. So they could see two they could see the pattern in wild-type cells, and in the same experiments, they could knock out another gene that affects that pattern, and they could see change in the structure of the neurons that correlates with the change in the localization of the protein of interest. And they also demonstrated they could do double knock-ins to label 2 different proteins in the same cell. It's a beautiful paper. Came in Cell in May, I think. And with that, I'm coming to the end of my slides, and I'm exactly at 1 hour, so that's pretty good. Just to summarize, so what can we do? I think you're all convinced by the end of the course that whatever you were thinking of doing before you came here is possible, and just go home and do it. It's very likely to work. In terms of making mice, I think at this point it's completely unnecessary to go through all the trouble with making ES cells, with some exceptions. So everything I showed you was oligos, and we've done things with small insertions. Bigger insertions still remain quite problematic in mice, but not impossible. So that might be one reason to go back to ES cells. Of course, you will go ES cell route if you also want to study ES cells, and that's still a useful reagent, right? But it is very fast method because almost always we get F0 transgenics, then very rarely mosaic, so you could actually study the founder, but of course, usually you don't want to do that before you breed them. But at least if you really urgently must look at the phenotype, you could do it. Takes less than 2 months. Basically, it's the time for development, plus a day for injection, and some time to prepare the reagents. Almost everything transmits. This has always been a problem in the past. Here, we don't even worry about it anymore. Then efficiency can be very high, as I showed. We had some litters where 8 out of 8 mice were edited, but of course this varies a lot. This is an important point. Varies between individual litters with the same mix, so there is also some randomness to it, but also varies significantly between different loci, different genes, right? Depending on the local chromatin context, probably, or some other issues with accessibility, or specifics of the, heteroduplex formation or whatever of the specific guide. We don't really understand everything yet, but we have seen that for the majority of the genes we always do 2 injections as a minimum, but often only one of them is enough to give us founders. But we have had really stubborn proteins. We had to inject like 15, 16 times to get a single animal. Luckily, the small minority. This was like 1 or 2 proteins so far. Occasionally you get biallelic, but it's very rare. More likely to get big deletion on top of what you want. So watch out for that. I covered it already. And finally, what the WAS slides were for, in the future, this in situ genetics, this ability to just take an organ. And of course, you wouldn't be able to do that everywhere. It's important to stress that it's probably going to work well in developing tissues or tissues that have actively cycling cells, because homologous recombination specific is not very efficient in non-dividing cells. But you can do it in the brain. You would be able to do it in other tissues that you can electroporate. There are some limitations, which I covered. I'll just summarize again. And there are ways to address them. So the PAM is one thing. As we know, it's required, and it's not everywhere, so that limits where you can do edits. But there are already Cas9 variants that have relaxed PAM specificity, or there are other Cases that have different PAM specificity. So that the toolkit is expanding, and soon practically everything would be possible. I don't know if you've noticed, but there is an Argonaut. There's a DNA-guided Argonaut that doesn't require a PAM. I don't know in practice how useful that would be. I haven't worked with it yet, but
so
are you working with it?
Hundreds of labs have tried it, and nobody else has had any
Really?
success. We were gearing up to try it, so we've stopped now.
Oh, that's some bad news.
Yeah.
This paper looked suspicious because it went through very long review, but
Yes.
but it is still
So we're holding
very
on for
interesting.
now, but,
Yeah, we started making the protein, so maybe we should.
well, let us know.
We'll do that.
No, it sounded great.
So anyways, even without it, we're looking good at this point. Off targets, as I said, can be diminished by a number of mechanisms. I list them again, and I just add two things I didn't mention, but you probably talked about, like nicases and uses of that Cas9 linked to FOC1, which would be kind of going back to the old zinc finger and talon idea, but this time with Cas9. This is being used. All of these things are way too complicated for creating models, where you can basically afford to discard the ones that are not what you want. But if you're going to, then if many people think about genome editing in humans to resolve genetic diseases and so on, of course, this is not something I am into it, but I realize that in that case, there's no room for error, so this might be important there. Anyway, so finally, the size of Cas9, it's a really significantly large protein. So this Argonaut was much smaller, but there are other Cases that are a bit smaller. And I think this is mostly a problem for viral vectors and other things where the amount of DNA you can insert in a vector is critical. If you're injecting protein, it really doesn't matter what the molecular weight is, as long as it gets to the nucleus, right? And that's it. That's all I have. I want to acknowledge many people, because I showed work from many years, especially in the beginning, all that stuff. And I tried to acknowledge some of the people on the slides, but obviously the Modern Code project was a gigantic consortium, and many people contributed. And there's a former and present members of the lab. And I want to point out, if you guys are looking for a next job, we're hiring. And that's it.